Welcome to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Martha Echevarría, and I am a postdoc at the University of Wisconsin-Madison. And today I will be moderating with Palominos, a postdoc at UC Berkeley. We're very excited to highlight the work of our outstanding postdoctoral members. And today, Joaquín Navajas Acedo from Biozentrum at the University of Basel and Yiqun Wang from UC San Diego will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of questions and answers. Please enter your questions in the Zoom Q&A box. Today is a pleasure for me to introduce our first speaker, Dr. Joaquín Navajas Acedo. He possesses an extensive amount of scientific training. First, he completed a master's at the Cajal Institute in Spain under the mentoring of Dr. Juan José Garrido, where he studied the role of purinergic signaling in the axonal cytoskeleton of mouse neurons. After this, he pursued his doctoral training at the Stowers Institute for Medical Research in the laboratory of Dr. Tatiana Piotrowski, where he explored the role of Wnt and PCP pathways during the establishment of hair cell orientation in the lateral line of zebrafish. Currently, he is a postdoctoral fellow at Biozentrum at the University of Basel in the laboratory of Dr. Alex Cheer, where he studies the somatosensory system of the zebrafish. His research has been published and highlighted in numerous prestigious developmental research journals such as DevCell, DevBiology, Development, Nature Communications, among others. Additionally, he has been awarded multiple fellowships and awards, including the 2025 SDB Trainee Science Communication Award. And today he will be presenting a talk titled The Spatiotemporal Emergence of Somatosensory Neuron Diversity in Zebrafish. Go ahead, Joaquin.
Sorry about that. Thank you, Martha. Thank you for the introduction. And I'm very happy to be here. Thank you for the invitation to present my work. I'm Joaquin. I'm a postdoc in the Shear Lab. And today I'm going to tell you about the ongoing efforts that I have to pursue a question that I'm really, really interested in, which is how does the nervous system develop and while it develops, create the many, many, many kinds of neurons that we can observe. Because take this drawing by Cajal of the human retina. This is literally your brain, and there are many, many kinds of neurons. They all look different. They all have different functions, and they all have in different parts of the nervous system. And they have to be at the right place at the right time and in the right proportion in order to create a functional tissue. So it is my main interest is trying to understand the molecular and cellular mechanisms that allow this while the embryo develops. Because you're going to see just in a moment, it is it's a very, very complex thing because different animals have found different solutions to the generation of cell diversity. Even different parts of the nervous system use different tricks. For instance, one thing that you can change or you can use is how you divide, right? You can divide symmetrically, right? Like progenitors and create more progenitors. You can divide symmetrically and create two neurons or two glia, or you can divide asymmetrically and generate one neuron of a kind. It's also interesting that where they are also influences the kind of neuron that you create, right? Positional information within the animal, so humor anterior, humor posterior, or for instance, a classic example of this is the dorsal-ventral patterning of the spinal cord, that depending on the kind of levels of hedgehog or sonic hedgehog or wind that you see, you're gonna develop into a neuron A or neuron B. But also not only where you are, but also who your neighbor is. So if you're lucky and then your neighbors are kind to you, you get to be one kind of neuron, or you get to be another kind of neuron. So for instance, one example of this is notch patterning, right? Like your neighbor and you enter this tug of war on signaling, and in the end, this creates very stereotype, or can create very stereotype patterns, such as, for instance, the inner ear, or in other organs. Not only where you are or who your neighbor is, but also sometimes animals include time as a variable to create diversity. So for instance, when they divide, so classic example is in the CNS of Drosophila, that you can have a progenitor that, depending on where and when it divides and the transcription factor that it expresses, is going to create a specific kind of neuron in the right proportion, right? But then there are other times where not only when you divide, but also the probability of you dividing and creating a specific kind of neuron is what generates the diversity in a tissue. So for instance, a classic example is the vertebral retina, right? You have temporal competence on progenitor that is going to express a number of transcription factors, and probabilistically it divides and produces a cone or a rod or other kind, right? So all of these things are enough to generate diversity in the nervous system, right? But as you can imagine, this is a very complex thing to study because, for instance, just in the mammalian cortex, there are 17 billion neurons, right? So how does the nervous system create or integrates all of these molecular and cellular mechanisms to generate diversity? So for that, I took a reductionist approach, and I am studying a population of neurons that I think are simple enough yet complex enough to tackle this question. So these neurons are called Rohan neurons. They're called Rohan neurons because they got described by Rohan and by Beard independently. But actually, fun fact, they got scooped by Sigmund Freud of fame by 10 years. He described it as neurons in lampreys. But the interesting thing about these neurons is that they live in the spinal cord. They're very easy to image because, like when in zebrafish, to transparent, and you get a lot of embryos. They develop very fast. And for a long time, people thought that they disappear, but sort of by accident, I found that this is not necessarily true. And then they all stay until juvenile stages. And I'm using these neurons because these neurons are part of the somatosensory system. So they live in the spinal cord. So this is what you see in the spinal cord. And as they develop super early. And as they develop, they send these projections to the periphery because their function is to detect stimuli and peripheral threat, because they send signals to the brain and allow the fish to escape. And they do this because they, like I said, they have these beautiful projections, and they express specific channels and signaling molecules to allow this somatosensory function. And we know, from previous studies, including, I think Alvaro suggests this in the audience, what they express, but when I started this project, it was not completely known what this complexity was. So one thing you can do to study nowadays, to study complexity of a population of cells, is to do single cells. So this is what I did. And what I found is that they were transcriptionally heterogeneous, and I found more combinations or even genes that were not described in the literature. So for instance, while there were some genes that were shared between clusters, for instance, like this P2X3 APS and TRPV1, we found, for instance, mutually exclusive expression, in NTRK2 or NTRK3, and even neuropeptides, right? So there were more things expressed than what we previously known, including also transcription factors. The other thing that was interesting was that while there were some genes that are expressed in all Rohan neurons, so for instance, this piezo, which is a piezo2, which is a mechanosensory channel, there were some of them that were along the AP axis, but not in all of them. So these are subtypes, right? And then they're mutually exclusive. And interestingly enough, that there were some of them that seemed to be regionally restricted. So for instance, ADCY1A or proencephalin. So ADCY1A seems to be more anterior. Proencephalin is always in the middle part of the trunk, and TRPA1B at 24 hours is on the posterior part of the trunk. So this maybe suggests that there's a functional relevance to this. But because this was only looking one or two genes at a time, instead of doing that, I decided to look at many genes at a time. And I decided to apply this new Wee-Murfish that recently came out, which is basically you can take an animal, you embed it in acrylamide gel, and then you can do spatial transcriptomics, which is basically you detect one or two genes, you image it, you strip, and then you can do this for as many genes as you want. And Yiqun following later, she will go deep into, she will show you more of this. But basically what I found is that you can look at many genes at the same time. And like I said, you can do this for as many rounds as you want. And then in the end, you can sort of have a good idea of the complexity of these population of neurons. So while, like I said, while there are functional genes are in all Rohenberg neurons, there is a major split in functional split in them. So you're either NTRK3 positive or you are NTRK2 positive. So they're mutually exclusive. And then within those, you find specific subtypes, right? So within the NTRK3 lineage, you will find the ones that are only NTRK3 or the ones that are TRPA1B, like I said, in the posterior part. And then within the NTRK2 positive ones, you will find subtypes such as ADCY1A or proencephalin. I mean, this is too complicated. This is basically how it looks like, right? So while all of them are PSO, then you're either one or the other, NTRK2 or NTRK3. And then within those, you will find subtypes. The interesting thing about this and how it develops is that if you look at all the subtypes, they do not appear during development all at the same time. So this is progressive. So the beginning, around like 12 hours, you have no functional classes. You have a lot of transcription factors, but no functional classes. Then progressively, you start finding more and more. And today, I'm gonna talk about this, how you go from NTRK2A to NTRK2A and 3A, because if you look at one animal, for instance, if you compare the left side and the right side of the animal, for instance, is not symmetric. There's variability. And even actually, if you look at different animals, they all look different, even within and among individuals, right? But if you count how many are NTRK2 and how many are NTRK3, it's incredibly robust. So there is variability, so there is individual variability, but then there has to be some sort of rule to do this, right? So then the question is, what are these rules, right? Like, for instance, like I mentioned in the introduction, is a cell behavior related to becoming, one or the other, like a symmetric cell division, etc. And how are these numbers controlled molecularly that make them so robust? For instance, are these pattern products of one progenitor that divides with different probabilities into one or the other? Is it two progenitors that are intermingled, right? And in the end, end up with this pattern, or is it a little bit more complicated, right? You have one progenitor that divides into two, and then there's some refinement, right? Because we know that Rohenberg neurons are born during gastrulation, right? From previous work, but we did not know the lineages because this was done with techniques that didn't have the resolution. So in order to reconstruct the behaviors completely, what I did is live imaging. So I put a transgenic zebrafish in the light sheet, and it took a while because these movies are gigantic. There's 14 terabytes, and they're very difficult to manage. But if you do get to do that, you can see the entire process, and you can follow every single step of the development. And in the end, you end up with these lineage trees. And the surprising thing was that they're incredibly simple, right? So in comparison, another neuron that is born around the same time, such a motor neuron, divides 4 or 5 times until the terminal division. But it looks like Rohenberg neurons fall into two flavors. Either I see them dividing or I don't. But the interesting thing and the important thing is that I never see a progenitor dividing into two raw Harveyians. So it looks like each one of them arises independently. They move coherently and they do not mix or switch positions, or even the right side and the left side, they don't cross. So then the question is, because they fall into these two flavors, is it tempting to speculate, is this what determines this, NTRK2 and NTRK3 split, right? Because you can have the same lineages that I've shown you, and then a positive correlation between division and not division. So, is it lineage dependent or not, or not, right? So what I did is you can image the animal, you can fix it, and then do a spatial transcriptomics on top of that same animal to get both the lineage and the, molecular profile, right? So, this is how it looks like. So at the end of the video, you can identify the raw Harveyians, and then you can focus, for instance, in, these 5. And then if you look at what they are molecularly and whether they divided or not, there doesn't seem to be any correlation between dividing or like being, one class or another. And in fact, you can block cell proliferation using this hydroxyurea and acetylcholine super early, even before gastrulation starts. Raw Harveyians somehow form, and they still segregate into these two categories, right? So then the question is, how do they segregate if it's not linked to lineage, right? And the clue came from development, right? Like the development seminar. So of course, we're going to talk about development. The interesting thing about the NTRKs is that at 24 hours, they're mutant exclusive. But if you look a little bit earlier, there is a hybrid state between NTRK2 and NTRK3, right? So there is, all of them are NTRK2, and then some of them express NTRK3. And we know from previous studies that this expression of NTRK3 is depending on a transcription factor called RUNX1. So if you have a mutant for RUNX1, this switch doesn't happen. I have data showing that if you put RUNX1 extra RUNX1, you get extra NTRK3. So it's all on this expression of RUNX1, right? It's all on this expression of RUNX1 in some of them and not in all of them, not in others. The other interesting thing that happens after RUNX1 expression is that this expression is transient, and then RUNX3 takes over and is the one that keeps them as NTRK3. So I have mutants for RUNX3. If you mutate RUNX3, what they do is to switch, but then they switch back to NTRK2. So you need to actively maintain this. But this pattern forms because of RUNX1 gets expressed in some of them. And the coolest thing is that if you look at how many flip at the beginning is 50-50. It's not 50-50, it's close to 50-50. So it looks like it may be a stochastic. And it's not only stochastic in the number, but also on the pattern. So if you look at the real data, right, how many are 3A followed by 2A, or 3A, if it's 1 and 3, 1 and 2, and if you look at the distribution compared to a random distribution, it looks like it's in random order. So then what turns RUNX1 only in this number and, like in a stochastic order? So I underwent a CRISPR screen. I made many mutants, but today you're going to see only 2. Because I've targeted transcription factors that were expressed in raw Harveyians as specifically as possible, and at different levels, right? So the ones that were everywhere, the ones that were specifically to a subclass, or the ones that were only anterior or posterior, and so on and so forth. So one transcription factor that is expressed everywhere is islet1. So islet1 is a transcription factor that, when mutated, what it does is sort of blocks the maturation of the raw Harveyians. So they do express piezo, they do express NTRK2, but they flip less. So you can go from this to this, right? So islet1 is necessary for this ratio of expression of RUNX1. The other family transcription factors that is expressed with islet-1 in all rhombineons is this PO4 factors. So you can see PO4F4 is one of them. It is expressed in rhombineons, but not in motor neons, right? Which are also labeled by islet-1. There are two PO4s that I made mutants for, or three, but today you're going to see two, PO4F4 and PO4F1, which are paralogs of each other. And if you make double mutants and you get double heterozygous and you do an incross, the interesting thing is that it seems like there's a dosage-depending effect on RUNX1, number of RUNX1 rhombineons. So if you have two copies or more, you flip or you switch to to this ratio. If you only have one copy, so you have homozygous for one and then heterozygous for the other, you do it less. But if you have no PO4F1 and F4, you don't flip. So you have 0 RUNX1. So all of them will remain as NTRK, as NTK2. So this suggests that maybe there is a dosage sensitivity to the process. So what I told you today is that rhombineons, they get used as discrete neurons, and everything that happens for the diversification is post-mitotic, right? Somehow, transcription factors are expressed everywhere are enough to do this stochastic segregation. You may be wondering, if it's something as notch. I have notch mutants, they still segregate. So it looks like it all boils down to these 3 transcription factors. So our model right now is that in the ones that do not switch is because PO4F1 and F4 somehow do not reach a critical level to activate RUNX1, but then in the ones that do switch is because this PO is somehow sensed to activate RUNX1. After this happens, there is a deactivation of the NTRK3 program, and then RUNX1 stops being expressed, but in the meantime, it has activated RUNX3, and then this maintains the fate. And then there's this patterning in space, what I call the spinal cord module. Basically, the spinal cord telling each rhombineon where they are in space. So this will further diversify the subtypes. But then the question is, what comes next? And I'm very excited because these are things I want to pursue in my own groups, hopefully someday. So for instance, so how do these 3 transcription factors activate RUNX1 stochastically? How does the genome make the interpretation? Is it a threshold? Is how they bind? Is islet-1 changing a property of the chromatin? We don't know. And what else is helping producing this ratio? Because it's so sharp that it may be a stochastic in the pattern, but it may also have help with other non-cell autonomous mechanisms. Once you have this, how is this change between RUNX1 and RUNX3 to maintain the fate or to activate and maintain the fate happen? And how does RUNX1 maintain the fate? Because remember that I told you, if you mutate RUNX3, they revert back. So you need to actively maintain this fate, and RUNX3 is one of the genes that does it. And then the other thing that RUNX3 does is to shut down the NTRK2 program, right? Because they stop being NTRK2 and then expressing all the genes. So how does this happen, right? And then this patterning in space, this is one of my favorite questions, right? How do all these transcription factors sum up to generate all these anterior, middle, posterior, subclasses? And how is all of this translated into the molecular, cellular, and circuit level properties of an animal, right? Because there is, of course, a genetic component, and there's some sort of ground rule. So on average, all animals look the same, but then each one of them has a specific property and a specific ratios of each one of the subtypes, right? So how does this translate at the level of the circuit? And can we change properties to manipulate their behavior at the molecular, cellular, and the circuit level? So with that, I would like to thank you all for being here today. And everybody in the shared laboratory, especially Alex, for being a great mentor and getting with me and, being patient enough to talk about and brainstorm about the raw neurons. Everybody in the shared laboratory and, everybody who's helped us, collaborators here in Basel or across the world. And with that, I would like to thank you again, and I will be very happy to take any questions that you have for me. Thank you.
Thank you, Joaquin, for an exciting talk. I do have one question while I give some time to the rest of the people to type some in. So I know that you did your CRISPR screen and you focus into transcription factors. I was curious if those were the top two, if you have other players in mind, and if they were restricted to specific domains of the axial skeleton.
So that's a great question. I can tell. So I made. So these are not CRISPRs. These are like proper CRISPR mutants. So like basically like 2-3 kilobase deletion to to be sure that there was loss of function. I can tell you that I made a lot of mutants. I made a lot of double mutants. And I made a lot of mutants and double mutants with no phenotype molecularly.
Okay.
They may you have another mutant, but basically it's been very. I mean, it is what it is, but it's been very frustrating because I have a lot of mutants that I was sure that they will have a phenotype because it was a transcription factor that was very, very specific. I had no. For instance, it didn't modify the ratio, but that doesn't mean, for instance, that it won't have an effect in other properties that the neurons have, like their axon arborization or the connectivity, or it will have a behavioral effect, but it's something that I have not checked yet.
Okay. So we have a couple questions. So the first one is from Amartya Mitra, and her question is, are there functional differences between NTRK2 and NTRK3 expressing neurons, and if their physiology has been explored?
That's a great question. So there are actual physiological differences between NTRK2 lineage and NTRK3 lineage. So for instance, the RUNX1 mutants lose the NTRK3 lineage completely because they only lose the part that is NTRK3 dependent. So for instance, they also lose the TRPA1B. You can poke them, which is detected by piezo, and then they poke, like they do this very stereotypical thing, which is, C bending. So, basically you poke them, they do this, but they lose detection of mustard oil, which is depending on TRPA1B. So it's, there are differences at, probably the physiological level, maybe at the electrophysiological level, but then I don't think that people have looked at it. People have looked at raw neurons in frogs, which also have raw neurons, and then they have basically thrown many chemicals at them. But apparently, they're very boring. Like if you do GCAM, because basically they integrate and they have a single spike, and then they don't have a complex pattern of activation.
Okay. Our next question is from Thomas Drysdale. Does your single-seq data show significant initial variability in the levels of the two PAU4s between cells?
No, I have done single molecule. I have done HCR. I have tried antibodies, but they didn't work. But basically, all neurons, effectively, at least to me, they have the same levels of islet and PO4F4 and PO4F1 at the RNA level. The question is at the pro level whether that will be different or not. But it's a good question. Yes, I will have to tag actually the PO4F1 and PO4F4 and then measure it bona fide protein levels to sort of get to the bottom of whether there's any the differences at the protein level or not. Yes.
Our next question is from Alvaro Sagasti. Did you see similar subtypes and processes occurring in other sensory populations, such as the trigeminal and the DRGs?
Yeah, so Alvaro, we need to talk about this someday. But so the raw neurons and the trigeminal, they look a lot like each other. There are many genes that are only trigeminal. There are many genes that are only raw neurons. They like for instance, NTRK2, NTRK3, piezo, TRPA, all of these are in the trigeminal. The only ones I have not found were the peptidergic ones, the ADC-YAP and then procephalin. Those seem to be more specific to the spinal cord, and of course, in other parts of the brain. But yeah, like the rhombineons seem to have specific genes are somatosensory, but then they also have other genes are in other sensory neurons, such as the hair cells or the trigeminal, even like olfactory sensory neurons, like potassium channels are like in other parts.
And one last question. It's from an anonymous attendee. You show some genes that had a spatially organized expression pattern, anterior versus posterior. Is there any evidence for Hox function in further subclass diversification?
So the answer is no. There is no proof that the Hox genes are implicated in this, but I can tell you that in the single cell, one of the strongest signatures for subclasses is Hox genes. So I can tell you exactly where a rhombineon is because it expresses, Hox8 versus, Hox12 or something like that. But it's something that I want to do, right? Can I trick a rhombineon to think an anterior rhombineon to think that is in the posterior part of the spinal cord, and then does that change anything about the physiology or like the morphology, something like that? Because in the end, I think that everything that I shown you is related to that, right? Like this, the formation of the circuit, and I think that is to a point rhombineons are equivalent, but then at some point the Hox genes need to kick in, and this is something that other sensory neuron or people that study other sensory neurons, such as the dorsal root ganglia in other systems, know that depending on where your axial level are, functionally you're different, or even motor neurons use Hox genes to diversify. So yes, absolutely, yes.
Thank you so much, Joaquin. We don't have time for more questions. And now
Thank
Fenya will
you.
introduce our second speaker.
Okay. So our second speaker today is Yiqun Wang from the Scripps Institution of Oceanography at UC San Diego, where she's a postdoctoral scholar in Didi Lyons' lab. Dr. Wang has made especially important contributions to our understanding of cellular differentiation and developmental trajectories during zebrafish embryogenesis. During her PhD in Alex Cheer's lab at Harvard, she helped reveal novel gene modules involved in secretion and their relationship to unfolded protein response transducers. Her work also contributed to landmark studies that reconstructed for the first time developmental trajectories and lineages, giving rise to neurons, muscles, blood, and many other cell types in the zebrafish embryo. Work that was highlighted by Science in its 2018 breakthrough of the year. So now, in an exciting shift from zebrafish embryogenesis to marine snails such as Crepidula, Dr. Wang investigates how mRNA localization and asymmetric cell divisions are controlled, integrating spatial transcriptomics, live imaging, and genome editing to better understand early developmental patterning. So welcome, Yiqun.
Thank you, Fenya, for that great introduction. So yeah, I'm Yiqun, and it's my great pleasure to present my work to you today. And in this project, I have adapted a advanced spatial transcriptomic technique into a non-model organism, namely the marine sleeper snail, which is drawn here. There we saw some very interesting biology during their early embryonic patterning process. Embryonic patterning is where cell phases are specified and spatially organized during embryogenesis. And over the past few decades, we have uncovered the major molecular mechanisms that orchestrate this process, such as cell-cell signaling, transcriptional regulation, and asymmetric cell division. But if we look across the broader tree of life, we start to notice that a lot of this molecular understanding actually come from a few quite restricted lineages. So for instance, within Balecteria, we have a lot of studies in vertebrates in the Deuterostomia clade, and we have our beloved arthropods and nematodes models systems from the Ichthyozoa clade. But if we move down to this largest clade in Balecteria, namely Spiralia clade, we start to see very little molecular understanding for how their embryogenesis works. And I think this is such a shame because this group of animal is incredibly diverse and interesting. As adults, they occupy a broad range of habitats, exhibit a range of body morphologies and behaviors, and a lot of them are increasingly recognized as good research models in specialized research fields. But what I find most interesting about them is that despite this great diversity as adults, these animals are all united by this conserved ancestral mode of early embryonic development, which means despite being so different as adults, as early embryos, these animals all look quite similar, and they all perform this stereotypical cleavage program, which we call the spiral cleavage. And that's also why we call these animals spiralians and this clade Spiralia. So during spiral cleavage, the embryo gave rise to embryonic cells in a very stereotypic order and at very stereotypic embryonic positions. And due to this stereotypicity, people were able to come up with a standardized nomenclature system to consistently describe the cell lineages and identities in the embryos, even across different Spiralian species. And there are two major types of cells in the Spiralian embryos. One is the macromere, which are bigger cells that are on the vegetal side of the embryo, and they are generally denoted with big letters. And then there are micromeres, which are smaller cells on the animal side of the embryo, which we usually denote with lowercase letters. And there are A, B, C, D, 4 quadrants in the embryo, and the embryo is roughly organized in this radially symmetric pattern. There is a very long history of studying spiral cleavage, probably because it's very prevalent and it's such an intricate developmental program. Here, more than 120 years ago, in the Journal of Morphology, Edwin Conklin published his thesis with a series of very beautiful drawings that details the cellular arrangements, embryonic structures, and some subcellular structures even in marine sleeper snail, Crepuscula fornicata. And in these drawings, he also included some of his observations of lineage tracing results, which established that many cells in the early embryo have deterministic cell fates. And in more modern days, people have performed more sophisticated lineage tracing with injectable dyes and fluorescent imaging, which have produced a very refined single cell level lineage map from early embryos of Spiralian species. A few species have now these kind of lineage map results available. Here shown is results from Ilianassa obsoleta, the mud snail. And if you compare across a few different species, a lot of time these lineage identities are actually conserved between different animals. But despite this rich knowledge in embryonic morphology, cell arrangements, and the cell lineage and fates, we don't have a lot of molecular understanding for what's going on in a spiral cleaving embryo. So for instance, are these lineages actually different in their molecular content? And what are some of the molecular markers and determinants of these lineages? So these are some open questions. And to address these questions and close this knowledge gap, I decided to use merfish, which Joaquín also talked briefly about. So this is a fish-based spatial transcriptomic technique, which would allow us to assay the transcriptional content of spiral cleavage embryonic cells without losing this intricate spatial pattern that they form. And in our lab, we use Crepidula ultrasolia as our model system. So this is a marine sleeper snail, and their embryos are about 400 microns across and about 300 microns from the animal to the vegetal end. We're using merfish, which is a single molecule fish-based method that's been widely applied in thin tissue slices on the level of about 10 microns in thickness. And in recent times, people have pushed this to 100 or 200 microns in thickness. This is an example merfish a mouse brain section from our collaborator Bogdan Bentu's lab. And since this is a single molecule fish method, we can see here in this blown up of this little window in the mouse brain, each dot is a single transcript, and they are colored by their gene identity. So this shows the high resolution of this method. So with our collaborators' help, we have adapted merfish to image whole mount marine sleeper snail embryos, and that's about 300 microns in thickness from the animal to the vegetal end. And for this particular project, we targeted 200 handpicked genes that are more likely to be involved in early embryogenesis. We also constructed the first reference transcriptome for this species and designed our probes against that reference transcriptome. The experiments were performed in the Bintu lab on a custom-built microscope coupled with a microfluidics device to be able to do the sequential hybridization imaging that Joaquín described to you in the last talk. So in total, I have performed merfish on 65 embryos across embryogenesis with a heavy focus on the early cleavage stages. After merfish was being done, we developed the pipeline to segment the cells in the embryos. So here are my actual merfish data with the cell segmentation mask on top. And because we know so much about how cells are arranged and about the cell lineages in these embryos, that allowed me to annotate the cell lineage identities in these embryos. And here are some of the broader lineage groups colored for your reference. But in reality, I was able to annotate every single cell in these embryos according to what people have known and according to this standard nomenclature, according to this lineage tree. So what this allows us to do is that for the first time, we can now map gene expression on this lineage tree that has been around for over a century. So for instance, here is the expression of brachyury on the lineage tree. This gene has been identified as a third quartet micromer marker. And here we're calling it 3Q because this is like, we are not specifically calling out A, B, C, or D quadrants. But you can see that the expression is in through little a, little b, little c, and little d. And remember, we talked briefly about this fourfold radial symmetry morphologically in the embryo. And it seems like this also carries through into the transcriptional space. So if I highlight these 4 quadrants in the lineage tree, you can see the expression pattern is basically repeating 4 times. And this also becomes obvious when we map back the expression onto the embryo that we acid in merfish. You can see that the expression is very much radially symmetric, organized by the quadrants. And not only we have this data now to allow us to look at on single cell level, what is the gene expression look like, we can also get to the subcellular level to look at how individual transcripts are distributed without the cell unit. And that allowed us to look to identify some interesting subcellular patterns of how genes are organized. So in these embryos, I'm about to show you merfish data on. So this is a 20 to 24 cell stage embryo. And I did immunofluorescence on it first to give us more cellular context to help orient in the embryo. So here are the first quartet micromer and its descendants. Here are the second quartet, and then here are the third quartet. And the macromeres are the bigger cells on the 4 sides. So when I pull out some lineage marker genes from our merfish data, we can see that different quartets of micromer cells are colored differently, showing that there are transcriptionally different cell types that correspond to their quartets identities. But I want to specifically draw your attention to the second quartet cells here. They are the 4 cells on the edge of the embryo. And hopefully you can see that these green transcripts that correspond to the transcription factor LHX5 is asymmetrically distributed in this cell. If I put the immuno and the merfish side by side, this becomes more clear. And interestingly, in this particular embryo, we captured the second quartet cells in their mitosis process, and they are slightly desynchronized, so we can actually see the entire cell cycle process within this single embryo. So here is a cell at metaphase, this is at anaphase, and this one is at cytokinesis. And if you look at the fish data and focus on the green transcripts correspond to LHX5, you can appreciate that it becomes increasingly polarized in the embryo as cell cycle proceed and eventually get partitioned into only one of the daughter cells. And this daughter cell corresponds to the 2q1 cell lineage. And indeed, if we look at our gene expression on the lineage tree, we can see that at the immediate next stage, the 24 cell stage, after this division has completed, we see that this LHX5 gene indeed becomes highly enriched in the 2q1 cell and not in its sister cell 2q2. So this shows that this particular gene become lineage restricted due to asymmetric RNA partition and not some fancy transcription regulation. And in fact, this represents the major mode of how genes become cell lineage specific in the early embryo of our species. This is another example for the protocadherin gene that becomes a marker for the 1q lineage. And the first quartet micromere 1q is produced at the 4 to 8 cells transition. So in this embryo, the cells are dividing and in anaphase, but you can see that CDH transcripts are already accumulated at one end of the cell that correspond to the future first quartet micromere domain. And in contrast, this red gene, DLX6, is going to become a marker for the second quartet. And at this stage, it is largely retained in the big macromere cells, waiting to be segregated into the second quartet micromere at the next cell division. So this is very cool to me. And it also points to this intricate subcellular organization of RNAs. And in order to really resolve these subcellular domains of RNA and see what RNAs are locally organized, I have developed a label-free RNA community analysis to discover without any a priori cellular label or subcellular labeling what are the self-coherent RNA domains in the embryo. So this is an example embryo that we did merfish on at the 4 to 8 cells transition stage. After performing the RNA community analysis, we identified a few major RNA domains. So these domains, again, these are individual transcripts colored by the domains they belong to. So within each domain, the RNA profile is fairly consistent, but between different domains, there are different RNA profiles. So we have the yolky part of the macromere separating from the cytoplasmic part of the macromere. And also within the cytoplasm, we have a specific domain in the center of the embryo that corresponds to the future first quartet micromere. And this is another example at a later stage. And here we can also see some prominent structures on the subcellular level that correspond to what we think are centrosome structures or the cortex-associated patches. And it turns out that not only the RNA profiles are different within these bigger subcellular domains, but also these domains represent a dense RNA localization. So the RNA density is higher there compared to the rest of the cell. And because of that, we can actually visualize these domains using a total RNA visualization method. So either we perform total RNA RNA fish with a poly-A probe, or we could also do live imaging with a protein that binds to a poly-A tail. So that is done by injecting mRNA mix that's encoding this poly-A binding protein that's been tagged with the amnion grain reporter so that we can track where all the RNAs are in the embryo. So here is the live imaging that we have acquired. So in this particular video, we also included red fluorescent protein to mark the nuclei and cell cortex structure to provide more context. But the green is the RNA, total RNA that we are interested in this embryo. And you can see they do form these large aggregates, and they have this kind of like a ring structure, dispersing, collapsing, and so on. So it's really represents this dynamic RNA organization throughout the development. And here are some snapshots from the movie, and we can see that the time scale of these structures dispersing and forming. And we have more detailed analysis to describe how this cell cycle dependent RNA shuffling actually mediates the RNA forming aggregates and asymmetrically partitions into their respective cell lineages. So all this observation brings natural questions about how is mRNA shuffled everywhere in this particular manner in the embryo of sleeper snails. And for that, people have some models for RNAs are transported in the cells, which generally involves transporting along cytoskeleton, and that also requires some kind of RNA binding protein to mediate the process. And the RNA binding proteins usually can recognize and bind RNAs in a sequence-specific manner mediated by the UTR sequences, which people also call the zip code. So the next steps are to really get down to these components involved in this potential RNA transport mechanism. What are the UTR zip codes that are in our system, and what are the RNA binding proteins mediating their localization, and what cytoskeleton components this process dependent on. Yeah, so this is basically it, and I would like to thank your attention and thank my PI, Didi, and the people in her lab that has helped me on this project, and also Bogdan, who helped establishing this technique to uncover this cool biology in this non-model organism. And with that, I will take questions.
Thank you, Yiqun, for that great talk. So in the meantime, while people are typing their questions, I have a question that this might be a little bit naive because I don't know anything about Spiralia, but I was wondering if this like asymmetric distribution of total mRNA and also like the LHX5 mRNA that you're seeing in the second quarter is something that you believe is conserved across Spiralia, or is something specific of Crepidula?
So I think this has some aspect of conservation, but also some aspect of divergence. I actually have looked at this RNA localization in a few species, not enough. I would love to look in more species. But for instance, I've looked at 3 snail species so far, and they all have very similar RNA organization early on. So this is embryo at the same stage for these 2 snail species, and their total RNA by poly-A fish is shown, and they are at a little bit different cell cycle phase. That's why the morphology is different. So this one is at the dispersing stage, so you can see a ring, but in the sleeper snail, they already condensed, so that's why you see a dot. But overall, they follow a very similar trajectory for RNA localization. But when I looked at a different Spiralia species that's a little bit farther related from the snails, the Cislak Bergia stephaniae, it has a different pattern, but the RNA is still localized. So from the animal pool view, we don't see much, like it's just kind of RNA dispersed everywhere. But if we look at lateral view, we can see there is a patch of RNA that is enriched on the bottom of the vegetal end of the cells. And if we just extract the bottom half of the embryo, you can see like really there is patch of RNA. That's not every single macromere there. So this is a very different localization pattern, but still localized. So I think the localizedness might be a more general thing among Spiralia, but where they localize the RNA might be different within different clades.
Okay, thank you for that question. I didn't anticipate that you would have like an entire slide for that. So there's another question from Anonymous. When do these embryos become transcriptionally active? Are these mRNAs that are maternally deposited, or are these like nascent mRNAs?
So that's a good question, and we have some evidence that the maternal loading RNA is the major player here in this early process that I have described, and the zygotic transcription seems to turn on after. What I'm showing so far is up to 24 cell stage, but the zygotic transcription seems to turn on after at least 30 cell stage. So that's also in the Spiralian world, there is a symmetry breaking point that is when after 24 cells, the 25th cell is born to break the radial symmetry, and that's believed to be the dorsal organizer of the embryo. So from there, I think signaling starts to turn on and zygotic transcription start to turn on. And we have some evidence that before that, the zygotic transcription is very low.
Thank you. So another question from Amartya Mitra. At what point in the spiral cleavage program do we start to see differences between species? Is there a phylogenetic signal there?
I would love to know that. That would require us to do a lot more of this kind of molecular study in more species of Spiralias. But currently, there are a few limitations. One is animal husbandry is not figured out for a lot of these animals. Increasingly, we're able to culture them in the lab. That's one thing. Another thing is to perform this kind of molecular studies, we need good genomic resources such as genome, transcriptome. When I first started this project, we don't have genome, we don't have transcriptome. So those are the infrastructure that we need to build up in order to do this kind of analysis. So yeah, I would love for people to have more interest in this organism so that we can start to build some of these tools to really do these comparative studies because of this really stereotypical development and the ability to map orthologous lineage very easily across species, I think this is a very good system for comparative studies.
Sure, thank you. So then the last question from Billy Swalla. Gorgeous images, Yiqun. It is amazing that most of the RNA is highly localized. What tissues are you looking at for differentiation?
What tissue? So one thing we haven't established is that this RNA partition and asymmetric localization actually contributes to tissue differentiation. So that would require some kind of knockdown experiment to establish functional relevance. But just by our knowledge for which sulfate lineage these cells contribute to, for instance, the brachyury enriched third quartet micromeres, they contribute to a lot of muscle tissues. And the first quartet micromere that has a lot of protocatenaries, they give rise to also some neural tissue, but also a lot of epithelial tissue that's highly ciliated. So there is some kind of correspondence for what transcription factor does and their functional relevance for later cell fates, but we don't really have a good functional causal link there yet, which is the next step to do. And the challenge there is that, of course, these are maternal RNAs, so CRISPR-Cas9 is not going to help. We really need something that specifically can clear specific species of maternal RNAs. We are actively trying like morpholino strategies, which turn out to be quite toxic for these embryos. So there are a few different things that we want to try. But yeah, that's a good question.
Okay, I think we won't have like time for the last question, but thank you, Joaquin and Yiqun, for your excellent talks. And also thank you to all of our attendees. This seminar has been recorded and it will be available on the SDB website next week. Please join us for our next and sadly our last seminar on Friday, May 8th. where we will have Jennifer Watts from Nationwide Children's Hospital and Ryan Hewlett from Carnegie Science. So thank you everybody for coming.